Good evening, everyone. Can everybody hear me? We are starting our third day of the Week of Awareness, and I'm very glad to see all of you joining today. So my name is Justine, and I was like the lead of this initiative. So I will also be your host tonight. And I will introduce today's speaker. So she was a really great help in the organization of this week. Suzanne is one of these persons with multiple talents, and I'm taking this opportunity to thank her for letting us using her really nice underwater picture, as well as being here tonight. And this picture, we were using it for our entire promotion of the week. So thank you again very much for letting us using it. And so as you can understand, Suzanne is a very active underwater photographer. She also leads the underwater photography club at KAUST, as well as being a diver instructor. And beside all of this, and probably more that I don't know of, Suzanne Rosbar is also a third-year PhD candidate in marine science at the Red Sea Research Center under the supervision of Carlos Duarte. For her PhD, she's working on the ecology of Trichadana giant clam holobiont in the Red Sea, with a special focus on its relationship with light. For this project that she will present today, however, she has collaborating with her colleagues Silvia Arosa and Cecilia Martin. The three of them brought together the expertise on microplastic and giant clams and examined the role of this charismatic reef organism as a potential sink of the marine plastic debris. Before coming to KAUST, Suzanne received her bachelor degree in biology and her master degree in biological oceanography from Christian Albert University in Kiel, Germany. And she worked on the autoecology of deep and cold water coral under future ocean scenarios. After this talk, and before leaving it to Suzanne, I also want to thank really much the presence of Professor Nikos Agis-Christidis, who will be our distinguished guest and who will lead a little bit the discussion after the presentation. So thank you again, Suzanne, for participating, and I leave it to you now.
Okay, it's the first time I do this, so this is the first time I'm giving a presentation like this, so I hope this will work out fine. Yeah, thanks for giving me the opportunity to speak today. Let's move on. So I will talk today about a project that was led actually not by me, but by Sylvia last year. No, I think in 2017 already. She is a PhD student in the same lab as I am, in the lab of Professor Carlos Duarte. And by that time, as it was her internship project, she collaborated with Cecilia, who is the marine plastics expert from our center, and with myself, as I'm working on giant clams, on this nice project on the microplastic uptake of giant clams in the Red Sea. So yeah, the most credit for this project should actually go to Sylvia, but I'm very happy to talk to you about this project today. So let's speak about a short introduction on marine plastic in general. I think when people hear about plastic in the oceans, they're mostly thinking of all these plastic particles floating around. The typical image that comes to one's mind are these giant garbage patches in the Pacific Ocean where all these debris, fishing gear, other plastic particles, everything that's broken up in the ocean is accumulating, is floating around in these gigantic gyres, and you can see them here on the right in that photo. And I think, yeah, that's like the most common thing that people think about when they hear plastics in the ocean. You can even see the diver swimming around here. However, actually, only 1% of the plastic waste that's entering the marine environment is ending up floating in surface water. So many of the plastic is actually taken up by biota, it's eaten by turtles, by whales, by everything that's swimming around. It's fragmented to even smaller pieces and taken up and sinking down in the water column, where it can be biodegraded by other organisms, by microorganisms, by bacteria. It can flow back to the coastlines, where it's landing on the beaches, as many of you, I guess, have seen before. It can accumulate in these garbage patches, however, but actually most of the plastic seems to sink down to the seafloor, where it's then buried in the sediments or ending up in different habitats here in the benthic And this is particularly interesting here in the Red Sea, as Chichia, for example, as she's working on plastic, was involved in a project also some time ago where they assessed the abundance of plastic fragments in the surface waters of the Red Sea. So they basically went out, I think, every other week to take plankton nets out with them, but instead of collecting plankton, they actually collected the plastic debris in the surface waters. And they found actually remarkable low concentrations in abundance and also in weight of plastic fragments in the surface waters here of the Red Sea. They were actually orders of magnitude below those that have been reported for other semi-enclosed seas, such as the Mediterranean. And yes, there are some things that make the Red Sea quite special and that could lead to that. For example, that the surface runoff into the Red Sea is quite low. There are not many rivers that end up in the Red Sea, so there's less import of plastic particles coming from the inland, and also the coastal development and tourism in this area is not as developed as it is, for example, in the Mediterranean. On the other hand, we all know that the waste management in this area is rather poor, and especially this suggests that there should be way higher inputs of plastic than what we actually see in the end coming up in the water. So the question is, where does this plastic end? And one very important hypothesis here is that it's actually ending up in the ecosystem, in the different habitats that we have here in the Red Sea. And that this biological removal could be a very important process in the mass balance of plastics here in the Red Sea, and explain where the plastic ends up in the end. Actually, so both the uptake of macro and microplastic has been reported for different marine organisms. Both can be ingested by marine biota. However, microplastics are more likely to enter the food chain, where they can bioaccumulate then and actually represent a quite severe hazard across a wide range of marine organisms. There are reports from plastic accumulating in seabirds, in crustaceans, polychaetes, in sea cucumbers, in corals, and also quite some research on filter feeders such as bivalves. Filter feeders are especially of interest here because, including the bivalves, they ingest quite a large amount of plastic. And this is because of their feeding mechanism, because in order to get nutrients and particles from the water that they can feed on, they inhale the water, and thus also the plastic particles that are floating around in the water. And these plastic particles can then get trapped in their gills, be transported to their stomach, where they then end up in the organism, and eventually even in us humans once we eat bivalves, for example. Here in the Red Sea, it makes sense to look into one of the most charismatic members of not only Red Sea coral reefs, but Indo-Pacific coral reefs in general, the giant clams. Not only because they're very obviously big, as their name already says, and because they're very prominent in the reefs here, also because in general they're among the biggest bivalves on Earth, as their name already says. The most interesting and the most special thing about those giant clams, however, is that they are mixotrophic, meaning they have two different modes for feeding. They receive their nutrition from the normal filter feeding that we already know from other filter feeders, from other bivalves, from black mussels to an oyster, for example, but they are also living in a symbiotic relationship with unicellular algae that are providing energy through the photosynthesis. So that makes giant clams in general very special, but as they are so big and are also filter feeding a lot, it's very interesting to look into their capacities and taking up microplastics here in the Red Sea. And from now on, I will talk about this paper that I was talking about, the project that Sylvia was actually leading on the microplastic removal of Tridacna maxima here in the Red Sea. And I hope I give you quite some insight into this, but if you have more interest or you want to know more in the end, then I'd like to refer you to this paper, which has been published in Environmental Pollution in 2019. But let me talk to you about the experiment now. So in fact, we actually conducted two different experiments. In the first one, we wanted to check whether the exposure to microplastic beads is causing any stress in the clam. So are they harmed by the uptake of plastics? Also, do they even ingest microplastics? Because this is the first study on giant clams and microplastics in general. And also if the ingestion is dependent on the clam size. So do smaller clams take up more plastic or less plastic than bigger clams? The second experiment was aiming to understand the abundance of ingested microplastic, if the abundance of ingested microplastic is dependent on the concentration in the water. So do the clams take up more plastic if there's more plastic in the water, basically? For that, we used two different size classes. We had clams in a size range of about 15 centimeter, which are considered juvenile or young adults. And this is because these are thought to usually obtain most of their nutrients and their energy from their endosymbiotic algae. So most of their energy is coming from the symbiosis and not necessarily from filtrating. And then we had bigger clams in a size range of about 20 centimeters that are adults. And those have shown to have a quite high rate of filter feeding activity in comparison to the photosynthesis. And they use that, as I said, to extract the food from the seawater. Here at Seymour, then for the experiment, we had the clams in big aquaria, and then within each aquaria, we kept them in smaller containers, actually in those 10-liter feeding chambers. And here on the left, you can see the normal feeding chamber. You see a clam here, and then we have a pump. And then for these normal chambers, you can see slightly here a mesh that's attached to the chamber, and that was just to keep the plastic inside. Because what we did is that we added the plastic to these chambers, and then it was in there for 12 days. So we assessed the influence of these plastic particles through the clams for 12 days. However, the water body in here is very small, so you don't want to leave the clam in here for a long time without water exchange. So this is why we had this mesh. So fresh water was able to come in from the outside, but the plastic particles were not going out. And then the current pump was basically to move the water, to create a current, and then to keep the water fresh. On the right side, we kind of have the same chambers. They're only closed. They don't have this mesh, and we use them for the incubations that we did to assess the metabolic rates of these clams. They have these see-through lids, so you can close them airtight, and I will go into the metabolic rates in a moment. For the entire experiment, we had 3 different controls, and that was very important because there are different factors that can also influence these disappearance or the uptake of the plastic particles. The first group of controls included 2 empty chambers with microplastics, so no clams inside, and this is basically to check how many plastic beads are just lost by being attached to the chamber. You saw that there's this net on the side. There is the pump, for example, so it's important to check how much plastic simply gets stuck to the aquaria without being added or ingested by the clam. The second set of controls included also 2 chambers with 2 dummies. Dummies in this case means bleached giant clam shells. So we had shells from previous experiments. And we took only the 3D structure, only the shells, and put them into these chambers, and also added microplastics in order to check which influence the presence of a 3-dimensional object has on the adhesion of plastics. So how much plastic is just simply stuck to the 3-dimensional structure of the shell, but not necessarily taken up by a living organism. And then as a third control, we had also chambers with clams, but without plastic, because as I said, we wanted to check whether the exposure to microplastic is influencing the metabolic performance. So if it would, then in theory, those clams without being exposed to plastic would do better than those being exposed to plastic. For the first experiment, we used 5 different size classes of microplastics, ranging from a size of about 50 millimeters to about 500 millimeters. And we added them to a mix where the concentration of beads was about 3 beads per milliliter. And then this is like the first main mix that we used. And for the second experiment, we used kind of the same mix, but we used different concentrations of these different particles. So we use about 1/2, 1/4, and 1/8 of the initial concentration to see whether the concentration at all of plastic has an influence. And this is important because many experiments use very high concentrations of plastics and try to assess the influence then on organisms. Sometimes concentrations that are not comparable to what's out there in nature, and then it's a bit complicated, or it's a bit hard to compare it actually to what would happen in nature if you expose it to conditions that are not natural. as I said, the main or the objectives of this entire experiment is to assess the total plastic removal by Tridacna maxima, by one of the giant clams here in the Red Sea. And by that, of course, we check the active removal, the retention by filter feeding. This is the common thing that people look into, like how much plastic is actually ending up in the gills and in the digestive system, how much do the clams take up. But the very interesting and new part of this experiment was that we were also checking the passive removal. So the possible attachment of those beads on this material to the shells. And we checked all of this through direct estimates. So we dissected the animals and explored all the tissues in the end for plastic particles, both digestive system and gills. We also screened the entire shell surface to detect any plastic that would be attached to it. And we also checked the fecal pellets to see whether, the clams have taken up plastic, but then just pooped it out basically in the end. For the metabolic performance, we're coming back to these chambers that I talked about before. So here on the left side again, as I said, they don't have this mesh, so they are airtight closed. And we have these mini dot logger in here that are measuring the oxygen content in these chambers. So you can assess the decline during the night when there is no light, the decline in oxygen, or the increase in oxygen during the day due to the photosynthetic rate of the clams with these sensors. And by calculating the ratio of the oxygen production with the oxygen depletion during the night, so the respiration, you can have a proxy on how fit and how well the clams are doing. And we also have a tiny pump in here to again move the water, so there's like no boundary layer around the clam. And then for a rather indirect estimate, we also took aliquots from these chambers with the plastics on different days. So as I said, we collected the clams' tissues and we also counted the beads in the end, but we also took samples every other day to check for the concentration of plastics in the water. So how much plastic is lost over time due to different factors. And here you can see actually a photo of Sylvia in the lab, who is cleaning the clams after the experiment. She's washing off all the plastic particles that are stuck to the outside. So we were able to count them. And on the right, you can see me dissecting one of these clams to get the gills and the digestive system. And when we now look into the first results, which are the ones from these aliquots that I was talking about, so we basically just check how much plastic is still in this aquarium after a few days. We can see that there was a significant decrease of beads in the aquarium with a live Tridacna. Actually, the daily loss was about 13% of the beads. So from one day to another, 13% of the beads were not in the water anymore. By that, we don't necessarily know if it's just stuck to the aquarium, if it's stuck to the shell, if it's taken up, but it's definitely not in the water. In fact, 80% of the beads that we added in the beginning were lost after the 12 days. With lost, I mean we couldn't find them in these aliquots in the water anymore. However, we didn't see a significant difference between the two size classes, so we're ignoring this for the later part. When we then look into the chamber with the dummy, so the chamber with plastic and a clam that is not alive anymore, but that still has this 3D structure, we also see that there's a significant decrease in beads in these controls. The decrease here was even more severe. There were 22% of the plastic beads lost from one day to the other, and in the end, 95% of the beads were not in the water anymore. But when you look into the chamber without the dummies, we see that this decrease is way less steep. Still, there's some plastic left, about 4% per day, and in the end, 40% was stuck to something else, or it was just lost to handling or to being stuck to the pump, to the aquaria. But we see that the majority of the plastic particles was actually be lost in aquaria, either with dummies or with the clams, which means there is something going on with the clams that makes the plastic disappear in a way from the water column. We also see that there's a linear increase of plastic concentrations with the number of beads that are lost per day. So if there's more plastic in the water, also more plastic is lost during this experiment. So it's faster stuck to either the clams or the aquaria. And then when we look into the active removal, we also see that microplastics were actually found in all treated clams. So there was no clam that didn't contain any plastic. All of them had plastic in the gills, in the digestive system, or in both. In fact, about 100 beads per individual were found in the digestive system, and only a very minor fraction of about 1.5 beads were found in the gills. 80% of the clams retained all size classes. So there was no difference between smaller or bigger plastic particles. They were all equally taken up. However, in overall, this active uptake by the clams is only accounting for 0.34% of the beads of this initial concentration that we added. So the uptake by the clams is only explaining a very, very minor fraction of the lost plastic. Here we can see again that there was no real difference between the plastic particles that were taken up, the slightly trend to bigger particles that were a bit more often taken up than the smaller ones, but in general there was no real size differentiation. When we look into the fitness and the metabolic rates, we could see that the exposure to the microplastics did not necessarily lead to obvious changes in their behavior. So they could have closed, for example, more. That didn't happen. We also did not have any mortality events, so none of the clams died. We also didn't see any anatomical anomalies during this dissection, so no weird growth forms that happened, for example, but we saw a significant difference between the initial and the final gross primary production to respiration ratio, which is definitely an indication of stress in the clams under the microplastic exposure. And now we're coming to the very interesting thing, because actually 35%, so about 1/3 of the microbeads that we added were retrieved attached to the shells in the end. And this is even more true the more plastic particles were added to the water. So you can see in this right graph here, if there is more of the concentration of beads is higher in the water, so this is the highest concentration and the lowest concentration, we can also see that more plastic particles are added to each square centimeter of the clam's shell surface. Makes, in a way, also sense. If there's more plastic, it can also get stuck more to the surface. But the very interesting thing is actually that 1/3 is attached to the shells. And this shows us that not only the Red Sea Giant Clam, Tridactyl maxima, but giant clams in general can remove microplastics from the surrounding water. And they do this through two independent processes. First of all, through the active retention of the beads, so the uptake, the feeding basically on plastic particles, and that is something that we would have expected because it has been reported for other filtration feeders before, but especially through the passive removal, so a process which involves the attachment of plastic beads to the shell. This is very interesting because most studies that are published to date are only or mainly focused in on plastic ingestion, and then very few are considering the attachment of microplastics to the surface of marine organisms. And this is not only true for clams, because I told you before that this is the first study on those giant clams. This is especially true for any other marine benthic organism, and especially for corals, for example, because in many cases people only look into the uptake of coral polyps from plastic, but not if plastic could actually be attached just to the structure of the coral, even if the coral is dead and, for example, overgrown with turf algae. So for the first time, we were able to demonstrate that the passive attachment on these shells is representing the dominant component of microplastic removal in giant clams, but I guess in other organisms as well. And we see, or this is the take-home thing also, I think, for other organisms, that actually the shells of living animals removed more beads than those of the bleached shells in the dummies. And this is because of the microbiofilm, the epiphytes, all these tiny algae, encrusting algae, turf algae, or polychaetes that are growing on the outside of the shell. You can see two photos of shell outside here. You can see they're heavily overgrown with other things, mostly with algae in this case. And you can imagine that in this three-dimensional matrix also of sticky stuff, the plastic particles can get stuck very easily. And that's basically the same that you would have in a dead coral reef, for example, when the corals are overgrown with turf algae. That's a perfect matrix to get stuck. So our findings are actually supporting previous suggestions, for example, by Cecilia from this microplastic paper, that the massive coral reefs in the Red Sea could actually act as effective filters that are possibly dominating these passive removal processes and explaining the low plastic concentration in the surface waters, because the plastic sinks down and then gets trapped in the reefs, for example. And this actually lays the basis for further studies on other species. I think also corals, but as I said, they were very interesting here in order to understand how the interaction between the 3D structure of the organism and the microplastic is happening. And of course, these future studies should not only focus on one species, as we did here, but rather check how microplastics and, for example, a coral reef as a whole are interacting. And with that, I'm done for now, and I'm open for your questions.
If you want to ask a question, you can raise your hand and I will let you talk. Meanwhile, we already had a question on chat, and actually it's also a question that I had, so it's perfect. So Christina is wondering what would be the equivalent with the natural concentration of the concentration of beads that you were using for your experiments?
Yeah, actually, I don't have an exact answer to that, to be honest, because I'm not the microplastic expert. But I know that we went for very low concentrations already, because as I said, most experiments give very high concentrations. I'm not sure. I think the 3 beads might have been a bit higher than what is in the field, but that's why we went for the lower concentrations then. But we still found the plastic in the clams. And as I said, this is in this case, also a very preliminary study, so to say, because, for example, this effect of this passive removal is something that we didn't think about it before. So I think the main experiment started with, okay, we do just the basic plastic experiment. It's another bivalve. It's a very big bivalve, so maybe something is different. And then we realized, oh, there's actually a factor that nobody had thought about, but never really looked into. And this should, get more attention, I think, in the future.
Okay, no, that's really cool. So then we have another question from Luba. So does the low volume basin and the pump imitate the marine situation properly? Kind of the condition that you tried in your experiment.
No, of course, it's a lab experiment. You have to say that, of course, it is not proper. This is not what it looks like in the field. But what I can tell you, so there's no published data on that yet, but we actually went one step further this spring and took some of these results to the field, where we actually had a flume, which is a chamber where you can lead water through, and then added plastic particles in different habitats, not only in coral reefs, but also in seagrass and in different algae, to see what happens with the plastic in the field, in more field-like conditions. How does plastic behave? Is it stuck? And also in way more natural concentration, I'd say. But that's something for the next session, I guess, once we have the data on that.
Yeah, no, that's really cool, because I was also wondering kind of what will be the effect on microplastic on other corals, no? And maybe also see which organism will capture more beads compared to other. And so if we can see a different distribution of microplastic according to the species. So how it is dangerous to eat fish, considering the amount of pollutant in the oceans?
So I don't know how dangerous it is, and I think that most scientists are still looking into how dangerous it actually is. But I know that another student from our lab, Fadia, she did a study on plastics in fishes here from the fish markets in Saudi, where she collected, I think, all the common fishes that people would get here, and she found plastic in every single specimen that she got from the fish market. So not knowing what it will actually cause is not helping us at this point, but that's something that scientists will have to find out in the future, I guess. But the plastic is ending up in all these organisms. You can find it everywhere.
Okay. Yeah, it doesn't look really good. I have also another question, if nobody else has right now. I was also wondering, yes, if you can see, did you try to check how much plastic the wild type, the clam in the wild, in the Red Sea, are already having on their shell and compare it to your experiment? Like, did you try to extract the microplastic and see how much it was already retaining or not?
Yeah. So we didn't check for clams from the field with regard to the plastic that's attached to the outside. So Sylvia found definitely at least some fibers that we didn't, and so we added fluorescent plastic beads to the experiment. So we knew how the plastic that we added looked like and what we were looking for. And Sylvia found other fibers in the clams when she was looking for the plastic particles from the experiment. So it's most likely that there was plastic inside the clams when we got them from the field already. However, since we didn't, like, I think to be sure, the precautions would have been different. Like, we don't know if the plastic particles or the fibers in there are because we handled them, because we put them in the aquaria. Is that something they had taken up during the experimental period or something that they took up already before we got them from the field? With regard to everything that's stuck to the surface, well, the thing is, so there is no real assessment on these clams here in the Red Sea. And in other regions, they're all on the red list. And I already had to kill quite a few of them for my experiment. I always try to reduce the number to take as few individuals as possible. And if you want to check the outside, you would always have to take out the entire specimen, and you can't bring it back to the reef. If you bring it back to the reef, it will be eaten by anything else. So that's why you want to reduce the numbers. But yeah, it would definitely be cool to see how much is actually stuck on these ones on the field.
Yeah, okay. And I was also wondering if you were thinking of maybe trying to do a structure like of shell that you could kind of create and then put in the Red Sea to kind of try to filter, I mean, not filter, but capture, let's say, like the microplastic already present in the environment. Like if it was something that you were thinking about as an application of your research. Like, I don't know if it's possible.
No, I have never thought about that. But it's not, yeah. So the thing is, we also see that, like, it's not only the structure, it's also all these organisms that are growing on them. I think, like the turf algae and the polychaetes and stuff like that. So I think there's not even need to create like an artificial clam for that. But and that's something we unfortunately didn't have the time to do this spring when we did our field work. But that's something that I'm very interested in the future. Would be to compare, for example, with a coral, a live coral, for example, of one specimen and one that's dead, but it's overgrown by other organisms. Because, like in my theory is that people are always looking into the live specimen and then what ends up in the coral. But for example, the dead reef could maybe trap even more plastic because these sticky algae are overgrowing everything and could trap it, things even better. So by just simply like taking an overgrown coral piece and examine this, could already be a solution maybe.
Okay. We have a last question. So does the type of polymers influence uptake by clams? From Philip.
I guess so. I think so. The reason why we choose polyethylene in this case is because I think it's the most common plastic that are found here in the Red Sea. So actually fibers I think are even more common, but with fibers it's very hard to work. So if you want to do a lab experiment, you would want to have like a specific plastic that you can, like for example the fluorescent bead you can see under the microscope. That's very convenient. So I'm pretty sure that there are particles or different polymers that are more easily taken up as the beads that we take. Yeah, there's a difference.
Okay. Well, thank you very much, Suzanne, for this talk and like the interesting discussion. So, and thanks also for giving a little bit of your time. I would like now to introduce our distinguished guest, who is an emerite professor of KAUST, Professor Nikos Agikristides. So students for sustainability are very honored to have you tonight joining us. It's really kind. And so your research is mainly focusing on synthesis of novel model homopolymers or copolymers. And before joining KAUST in 2011, you were a professor of chemical science. Professor Agikristides was a professor emeritus at the University of Athens, while he received also the Chaired Awards as Excellence in 2018. So you can welcome him by clapping. You have like a small reaction icons when we can all clap hands. So we can all do that if you want. And
Okay.
I leave it to you. Thank you very much again for joining us.
That's okay. To tell you the truth, I am honored to be this evening with you, with the students, because I am always happy to interact with the students. The reason I accepted the invitation with very pleasure. And to tell you the truth, Susan, for me it's the first time I give a Zoom talk, which will be very, very short. So feel free to invite me anytime you have some workshop or some talks related to my specialty, which is polymer science. Okay. First of all, I would like to congratulate Susan for the excellent talk he gave a few minutes ago, and also the whole group, Cecilia, Sylvia, and my good friend Carlos, for what they are doing, because what they are doing is very crucial for the planet. So also I would like to congratulate the committee of KAUST students for sustainability. So I have instruction from Eman to give a small introduction to microplastics. And after 5 or 10 minutes, already the 5 minutes passed, and after to discuss with you and to answer to any question you have concerning the pollution of the environment by the plastics, okay? So first of all, let me clarify two terms, plastics and microplastics. So plastics are members of a big family. The big family, the name of the big family is polymers. So plastics are not the only members of this family. There are, for example, fibers, there are rubbers, elastomer, etc., etc. So this is wrong to speak about plastics. Plastics is a very small category of this big family by the name polymers. Okay, now what are microplastics? Microplastics are polymers that they have dimension less than 5 millimeters. So 1 millimeter is 1,000 micron. So it means that these materials, they have dimension less than 5,000 micron. So these micro plastics or micropolymers have nothing to do with the unit. We name microplastics because micros in Greek language means small. So that's why nothing to do with the unit, the physical unit, but this is small particles. This is like the grain of rice. Okay. Now, from where these microplastics they are coming? We have two kinds of microplastics. The first kind we call primary microplastics or micropolymers. These are engineered to be small. Why we do this? Since we need them in medicine, in cosmetics. For example, there are many microfibers in mascara. They use the microplastics for exfoliation of dead skin in cosmetics. Also, we are using microplastics, small spheres, to prepare in the extruders all the items, all the polymer items, all of them. So these are the first category that is coming because we need them, okay? So the second category, the second kind of microplastics are secondary microplastics. Those created by us, those created by the degradation of macroplastics, of the big bags, cups, bottle, and from the laundry machine, for example, by degradation, either this biodegradation or chemical degradation or mechanical degradation. So how we can stop the presence of micropolymers, microplastics in our environment? First solution, to stop producing polymers. This is completely unrealistic because polymers are in everyday life and they do many things. If you imagine how many polymers you are using every day, they save lives because they can be used as artificial organs, okay? And also, you don't think about that. They save environment. For example, imagine if you have a plane constructed, built by metal, or cars. These will be much heavier and they are going to use much more gas. So will be much more pollution. So this is unrealistic to stop producing plastics. Okay. Second solution is to clean the environment from the microplastics and continue to producing more and more every year plastics. This is also unrealistic. Why is it unrealistic? Because imagine that you are going after a job to the house. Now there is no lab, so we stay all the time at home. And your sink in your kitchen is overflowing. So what you are doing? You clean the floor? No. What you are doing, you close the tap of the water, okay? So the first thing we have to do is to try to don't use a lot of plastic. For example, we don't use single-use polymers, okay? This is a big mistake. And after we have to recycle, okay? And after, what we are going to do? We are going to produce, the scientists, smart polymers. What are smart polymers? Usually, the plastics or the polymers, they are produced from small compounds. The name is monomers. The monomers are coming mostly from the crude oil. So the monomer are giving the polymer, the big molecules, we are using and after we are disposing. This we call linear polymer economy. So imagine, and we started working on that we have monomers, we produce the polymer, we use, and after we get 100% monomer back. It's not recycling, it's just recovering the monomer, and we produce again the polymer. So the solution is to have polymers, production of polymers with no waste, okay? Now, let's say why I said cleaning is an utopia. For the moment, we have to do first the other or the same time. In order to clean 1/10 of 100th of the North Pacific Ocean, we need 70 boats to work 1 year. So with this small introduction, I am ready to answer to questions. And I would like to thank you very much. And again, I would like to thank the organizing committee for the sustainability at KAUST to invite me. Thank you very much.
Thank you a lot, Professor, for this very nice summary of what we could do and what would be the solution. Anybody has any question? What kind of research exactly are you doing in KAUST then regarding polymers and monomers?
Okay, this is a general question. What we try to do is, you know, that if you compare in order to, for example, you have the rubber. The rubber is, you cannot recycle the rubber. I mean, the tires. And why? Because it's chemically is cross-linked polymers. It means you cannot melt in order to reshape. You cannot dissolve. The only thing you can do is to cool down below a temperature. The name of this temperature is glass transition temperature. Below this temperature, the flexible tire becomes solid, like glass, and you can break, and you can mix with asphalt. And when you mix with asphalt, you can do the roads. But this is not the good way of using this, because there are so many tires that you cannot use all of them on the roads. So what is a part of our research is to produce thermoplastic elastomers. Thermoplastic elastomers means there are triblockopolymers. So there are, in the same polymer, there are two different chains, covalently bonded. So the one chain at room temperature is flexible. The other is hard like glass. So at room temperature, they behave like rubber. So they are like physically cross-linked. But on the other hand, at high temperature, they become again linear thermoplastic. So you can reuse, you can recycle it. This is the one part. The other part, what we are doing, we are using monomers from corn, for example, in order to produce biodegradable polymers. Okay, polycyclic ethers, or the monomer, the cyclic ethers. So we are preparing polyethers or polyesters. And the third part, I have one new postdoc and another one is coming, is we are going to working on this direction, preparing polymers without waste. So monomer, polymer use, and again monomer and polymer.
Okay, thank you very much. So we had like a few questions that pop up while you were explaining. So 3 of them from Christina and Mish were like very similar, sorry. So are you working on any type of polymers that will be more easy to degrade? So this is actually what you were mentioning in the last.
Yeah, I mentioned already, polyesters like polyester, like polycaprolactones or polylactides. They are very useful for you know, they have very good mechanical properties you can use, but after you can degrade very easily because they have the ester group.
Okay. Iman is asking, is biodegradability is a good thing, first of all? What do you think?
I think that the biologists, they have to answer, but all polymers are biodegradable. All polymers are biodegradable. The question is when. The one at the end, for example, polyethylene in 500 years is biodegraded, okay? The polycaprolactone in a few months. So first of all polymers are biodegradable, okay? So
if I can interrupt really quickly, I think this is what, for me, this what makes it really dangerous, because they're biodegradable. They will circulate in the living system. When they do so, they will cause other problems.
I did finish my answer.
Go ahead.
Okay, this is one part. The second part, I was following one talk today in the internet, was American scientist that he was working on PET, polyethylene terephthalate, to find the microbes that they can degrade this polymer. And PET is everywhere, okay? For example, all the bottles. Now, she's not using different microbes that there are in the environment. So what is doing, she's training some microbes to eat polypeptides because they PET, polyethylene terephthalate, because polyesters, because they need to eat for energy. So she's training not with new microbes, but just microbes that they exist, many in our planet. So I don't know if this is going to break the equilibrium that there is between, you know, microbes and human or animal lives. So now you can say what you started saying.
Thank you. I mean, we live in uncertainty. I think at this stage, we don't know if we're gonna be resilient enough or if it's gonna break the equilibrium. Do you agree? I don't.
But do you agree with me that
yeah, I agree with you.
we cannot live without polymers?
Yeah, everything is like, well, it's all around us. I cannot imagine my life without them.
So next time I can give a more extended, because Eman invited me 2 or 3 days ago. So, and I was really very busy with many things. So I just, you know, I thought that this small introduction maybe is going to clarify a few things. It goes like ballots, you know, what we can do, what we cannot do. But we have, I mean, this is the people, they have to understand that he has to, you know, they have to use one-use cups, bottles, and to recover all the plastics and to don't throw to the land or to the sea. Second, our politicians, that they have to understand that they have to put laws to the industry that, for example, the 30 or the 40 or the 50% of their production should be recycled, okay? And now all the polymer scientists, they try to convince them. And I don't like to think what will happen in a few years, like in these difficult days, because scientists, they were saying many years ago that nuclear power is not the enemy, it's a small virus, okay? So I think that we have to plan and to think about the future. And the third, of course, the scientists, they have to find the ways to prepare smart materials.
Yes. Thank you. We have a couple of more questions, if you have time, like maybe 5 more minutes.
I have time until tomorrow morning. I always like, I mean, this is much better than the talks, the question. So in my opinion, you have to ask again Susan. She's very good. So again, I mean, just we are here for questions. I am here. I don't have any problem to stay half an hour more.
Perfect. So then we have a question that is more directed to the chemical industry. So do you think that there is enough regulation for chemical industry to ensure the safety of market products coming from the chemical industry? So this is from Luba.
They have the capability. I don't know if they are ready to do, because there is a lot of money. I am a consultant of ExxonMobil. You know ExxonMobil, the big company, for many years now. So until a few years ago, the goal in the labs or in the industry is to produce stronger, better plastics to make money. Now it's the other way. It's the time arrived that we have to produce polymers that we can use again and again. The goal is different, because if we don't change this goal and to go ahead and produce better and better plastics without thinking about recycling or about the circular polymer economy, not the linear polymer economy, we are lost. In my opinion, they can use part of the the money they are doing, they make to this direction.
Yes, thank you. Because we were watching as well, like documentaries on Monday. I don't know if you saw like the Broken episode on Netflix about plastic production and recycling. And it was like quite concerning to see that in the end, most of these big companies, they are putting a lot of money to research new products, but not to actually try to solve the problem. They kind of rely on people to solve the problem for them and just continue producing.
To tell you the truth, now with the coronavirus, the politicians and the industry, the bio industry, they have to solve the problem. Now they cannot do anything else. Okay? So in my opinion, for microplastics, there are everywhere.
Yeah. Another question that I thought was really interesting and you didn't mention is regarding your research. Are you trying to make collaboration with algae biotech team, for instance, from KAUST, to make new type of polymers that are plant-based? So this is like asked by Mitch and also by John.
No, to tell you the truth, no.
And can I ask
for example,
why?
I will tell you what. For example, we are working on block of polymers. That this block of polymers is for selective targeting on cancer cells, and I was doing that in Greece. Block copolypeptides. You include a hydrophobic drug in a very smart vesicular structure, and is going to the cancer cell and is open, is delivered there. But this research, it's finished in Greece when I was in Greece. But now with these circumstances and this situation, I am thinking to start again, okay?
I was also wondering if you're thinking of, because you mentioned that you're trying to make new monomers, different type of polymers, and like think about the recycling of this new compound. But are you also researching or thinking of a solution of handling the plastic that is already produced and that maybe doesn't have any solution? And I was also thinking like, why the main goal of plastic recycling is to do exactly the same product and not trying to maybe make a new type of product with a different property. Like we don't have to replicate exactly the same thing.
Right. No. These are good thoughts. But imagine that you must have a polymer that is very strong to be resistant to UV radiation, to be resistant to oxygen, to be resistant to everything, to solvent, and after you like to degrade. This is not an easy problem. We need a lot of efforts. We need collaborations, but believe me, many scientists, they started thinking about that, and I am sure that the solution is coming. First, for example, polylactides. They can replace polyethylene in a few uses. And the polylactides, you can get again the monomer, which is lactide or lactic acid. Now, but it's not an easy task. because there are so many applications you don't even imagine. In one, you like to have flexible, and the other, you have to have elasticity, strong and elastic. You like to have good processability, but good mechanical properties. There are so many factors, so many parameters you have to take into account that, in my opinion, we need collaborations, and we are going to solve this problem. But this is one good direction, that you find another monomer which is friendly to the environment, is giving a friendly to the environment polymer, has strong properties, and after to be able to degrade to the monomer without waste, and after to do again the polymer. No, these are very good thoughts. I am impressed by this question. Yes, of course.
Thank you. We have another question that we didn't ask yet. So what happened with the additive after degradation? Are they released?
Yeah, this is a very good question. Okay. So the microplastics, I forgot to thanks for this question. Okay. So for example, you have the polystyrene cups. Okay. The polystyrene cups are flexible. Okay. The cups of coffee, the foam. Okay. So if you prepare the polystyrene in the lab or in the industry, this is like glass. In order to make flexible, you have to increase above 100 degrees, right? Above the class transition temperature. What you are doing, you cannot do that. So you are putting some compounds we call additives. For example, plasticizers, in order to make it more flexible. The plasticizers are small molecules, are going between the big molecules, and the intermolecular interaction, they become less, and so the TG comes down. So they become flexible. Also, in order, if you leave polystyrene in the atmosphere, maybe the life will be the life not to degrade to the monomer will be a few weeks. So you have to put a dioxidant. Most of them are chemical that the governments, they try to have this chemical not toxic, but a few times it's very difficult to avoid. So these are going out. They are going out, okay? And this is toxic for the fish, etc. But the most important is the following, that this microplastic is like a sponge. They take from the atmosphere, for example, the chloro aromatic compounds, toxic, okay? And they are going to the sea, or they are going everywhere. So this is a good question. The additives that they are inside the plastics, and the additives, are changing the properties. And that's why I told you that until now, until a few years ago, we were not thinking about this. We were thinking only just make the, for example, in the toothbrush, okay? In the toothpaste, there are nanoparticles, there are micro. But in 2015, the USA, just does not give the permission to use anymore this, okay? What else?
I don't know if we have any other questions.
I try to, yeah. And I am straight all the time.
Yes, no, it was really nice to have you here. Like the discussion was very interesting. I'm very glad that Iman contacted you. It was a great addition to today. So I guess we don't have any more questions, but thank you very much for joining us. So we have again event tomorrow from 7 to 8 p.m. We have a different link and it will be about recycling. Okay. So thank you very much everybody for joining and
Maybe
have a nice evening.
I will follow this discussion. Okay. Thank you.
Thank you very much again for joining. Bye.
Thank you so much. Thank you.
And thanks again, Suzanne, for your talk. It was really good as well.
Suzanne, it was better talk than mine. It was scientific, 100%. Congratulations again.
Thank you all. Thank you.
Bye.